Julian Paganini. I work for the Institute of Risk Assessment Sciences at Utrecht University at the Netherlands, and today I'm presenting some of the work we've been doing for a source attribution of Salmonella Typhimurium, and it's a comparative analysis of machine learning approaches to do that. Source attribution is basically a discipline that tries to predict from which source a particular foodborne pathogen comes from. And in this case, we are applying it to Salmonella Typhimurium, which is one of the most prevalent foodborne pathogens worldwide, but also in the European Union. Basically, Salmonella Typhimurium is a very complex pathogen to do source attribution because it's a host generalist, which means that can colonize and infect multiple animals, like for instance, pigs, cattle, and poultry, but also wild animals and sometimes aquatic animals. So the source attribution of Salmonella Typhimurium is complicated. In this particular work, we're using machine learning and we're trying different machine learning approaches to do source attribution. And luckily, these type of artificial intelligence approaches are very powerful for predicting complex tasks. Just to summarize, the research objective of this work is basically comparing different machine learning approaches coupled with different genomic representations of Salmonella Typhimurium for source attribution and trying to see which one is the best approach. So to start with the data and the methods, we took advantage of this big reservoir of publicly available data that we have, and we collected from public databases more than 11,000 Salmonella Typhimurium genomes. And one of the things that we first did was explore the source distribution and the continent distribution of this data. And one of the things that we discovered is that the sources are unbalanced. And that's expected because most of the surveillance sequencing being done in Salmonella usually points towards the most likely culprits of human infection, which in this case is poultry, swine, and bovine. And these compose the majority of the genomes that are publicly available. Aside from that, one of the other things that we discover is that around 80% of the data comes from high-income countries, mostly the US and European countries. So these available genomes we collected from two sources. First, we collected from Enterobase, and Enterobase we collected the source information, and we also collected core genome MLST alleles. And with the core genome MLST alleles, we did two things. First, we derived population structure features. So we define the population structure in two manners. We first define stable lineages, and these were defined by clustering the three using a 60 allele difference threshold. The way that we derived this threshold was basically clustering the three using all the thresholds between 0 and 300 allele differences, and we measured the similarity between the clusters at different thresholds. And we evaluated how similar the clusters were to each other. And when we found a region in which no matter how much we were changing the threshold, all the clusters remained kind of similar. So we find the first stability region at 60 allele differences. The other population metric that we derived were defining outbreak-like clusters using 5 allele differences as threshold, which is what most public health reference labs use. So with core genome MLST alleles, we also one-hot encode these alleles. So we obtain at the end a presence-absent matrix, which has as rows each of the 11,000 isolates that we are including in this work, and as columns each of the allele variants for each of the core genome genes. Aside from Enterobase, we went to all the bacteria, which is a great resource, and we downloaded the corresponding assembled genomes, full genomes from these bacteria. And what we did is we built a pan-genome graphs with them using a tool that is called Bifrost. And then from that pan-genome graphs, we were able to represent each of the genomes as a unitic presence-absence matrix that has a similar dimensions that a similar structure than the one that I explained before with core genome MLST. Each of these individual matrices were used as an input for a package that we developed to streamline machine learning workflows in microbial genomics, which is called Machine Learn. And here, in this QR code, if you scan it, you can actually go to the tool and try it yourself. It's very easy to install. It's a Python package and it's hosted on a pip repository. So the way that this machine learning Python package works is it follows a set of steps to train and evaluate machine learning models for any task prediction of classification in microbial genomics. And the way it starts by splitting the data set that you give as an input. And it creates by default, but this is tunable, a 20%, 80% division. So 80% of the data will be used for training the models and 20% of the data will be used for testing the models. But the way that this tool creates this split is aimed to address a very particular characteristics of microbial genomics data, and it's the fact that it's structured. So in microbial genomics, when you analyze a set of isolates, these isolates are not actually independent points from each other, right? Because they have an evolutionary relationship between them. So at the data set split, basically the tool does two things. It uses the population structure features to create a stratified separation in which the previously defined stable lineages are actually equally represented in both the training and the test set. And aside from that, and this is very important, outbreak-like clusters, which we also defined previously, are moved together either on the training set or on the test set. And that way, you avoid data leakage. That means you avoid training your machine learning pipeline or machine learning model with the same data that you're testing after. And this is a very important thing and a very common limitation in a lot of the machine learning studies out there in microbial genomics. So once the data set is split, this 80% of the data that we selected for training actually goes through a feature selection process. And the objective of feature selection is obviously reducing the amount of features that you need to use for training the model. And this is an optional step depending on how many genomes you have and how many input features you start with. And you can choose between one or two different feature selection algorithms. One of them is a simple chi-square test, and also the other one is a multivariate recurrent elimination of the feature, or MUVR. Once the features are actually selected, what we do is we train and optimize the algorithms. So at this point, at this moment in time, the tool, Machine Learn, has the option to select between 3 different machine learning algorithms. This is random forest, stream gradient boosting, and support vector machines. But also, it has the option to choose between 3 upsampling strategies. So no upsampling in case that your data set is balanced, randomize, upsample, and smote. And your training data will go to a cycle of basically training, evaluation, which is done using group cross-validation, and then optimization of the model via finding the best hyperparameters that underlie the functioning of the selected machine learning algorithm. And this is all done automatically by this package. So after this training cycle, you will end up with an optimized model. And this optimized model is then fed your test data, so that remaining 20%. And then you will get predictions for the source or for whatever you're training on your test set. And also you will get evaluation metrics on how the actual model is performed. Once you have trained the model, you can also include an external data set that you have, that you want to see how it works. So if you want to predict where the sources are coming from, you can also add to this optimized model an external data set to obtain predictions. Okay, so now to the results of source attribution based on using this package. So on the first block here, we are seeing the accuracy of each of the models. So on the y-axis, we have the accuracy metric. On the x-axis, we have the different model combinations, so combinations between machine learning algorithm and upsampling strategy. And then each of the blocks represents the different inputs that we give to the model. So that's core genome MLST alleles, unitics, or a combination of the two. So one of the things, so here in this plot, the gray dots actually represent the cross-validation false results. The black dot represents the average performance of the model in the training set, which is the average or the mean between all cross-validation false, and the blue dots represent the accuracy on the test set. By quickly looking at this plot, one of the things that we can notice is that by looking at the black dots, that the unitic-based models in general perform better than the core genome MLST models, but just on the training set. However, when you look at the blue dots, we can see that blue dot is the test set. We can see that the core genome MLST models actually perform better than the unitic-based models. And we actually see a big difference in performance between the training set and the test set with unitic-based models, which means that usually these models are being overfit just by using unitic information. What is interesting is that when we combined both types of features, unitics and core genome MLST, we see a better performance both on the training set and on the test set. And these models based on unitic and core genome MLST input are therefore seem to be working better for source attribution of Salmonella Typhimurium. So the second block is showing the F1 score for the test sets for each of the models, and again, each pane in this graph represents the input. So core genome MLST, unitics, or unitic and core genome MLST. So one of the interesting things here is that majoritarian classes, like for instance, poultry and swine, which in this plot are described with a blue and a yellow dot, actually have a good performance in all the models, although unitic and core genome MLST input is a bit better. And then minoritarian classes, as expected, like for instance, aquatic animal and other livestock sources, are actually performing very bad in all the models. So these are very difficult sources to predict, especially because the amount of initial genome is very low. So the main difference across the models that we see is basically dominated by the classification of two classes. One of them is wild animal, and the other one is bovine isolates, in which the later models, combining both unitics and core genome MLST as input, perform better than the other two models. So just evaluating the best models with a confusion matrix, we notice that one of the interesting things is that bovine isolates, so true bovine isolates, very often get confused with swine. And no matter which is the algorithm that we use, this confusion between bovine and swine sources is very common. So going to the conclusions, we concluded after this study that the best performing models actually use as an input a combination of both unitic and core genome MLST features. However, in none of the cases, accuracy went above 0.85, and this highlights the limitation of just using genomic information as an input for source attribution models. There are definitely some epidemiological parameters that should be included to have more accurate models, and maybe also some geographical information. The second conclusion is that the fact that bovine-derived isolates are very frequently confused with swine, despite bovine isolates being one of the majority classes, seems to indicate that these two animals share a common reservoir or they are able to be infected by very, very similar isolates. And that's why the models are having a hard time trying to differentiate between these two sources. So I want to thank you to all the people involved in this project, which is definitely a large cohort of people. My work is funded by Health Holland, and the results are a collaboration between Utrecht University, Thiagen Genomics, and the RIVM, so the Health Institute from the Netherlands. Thank you to everybody involved in this study.